Okay, so welcome back to this next video in which we are discussing cystic fibrosis and cystic fibrosis medications. Okay, right. So when I say cystic fibrosis medications, really a lot of what I'm actually going to tell you about are potential medications rather than medications which are actually used at the moment. Okay, the medications that are used at the moment are mainly symptomatic. They're about treating the bacterial infection. So there's a whole list of antibiotics which people with cystic fibrosis will be taking. Okay, we're not going to discuss those. What we're going to discuss is potential therapies that are not actually used at the moment, most of them at least anyway, okay, but which are potentially things for the future, okay. Some of them are in clinical trials at the moment. Right, so the first thing that we're going to talk about is in vivo gene therapy for cystic fibrosis, okay. Now, this is a brilliant idea. The problem is it doesn't really work at the moment, okay, and I'm going to explain the problems that there are with in vivo gene therapy and why it doesn't really work at the moment. Okay, so what is the idea behind in vivo gene therapy? Well, all of cystic fibrosis is caused by having loss of function in a single protein. You do not have enough CFTR function at all of these different cells, and that is what is causing the symptoms of cystic fibrosis. That's what causes cystic fibrosis. So why don't we just replace the gene for CFTR in your cells, okay? Why don't we just put in a new gene, okay? Then the cells can use that new gene to synthesize CFTR, and then they'll be fine. They'll perform their function normally, and the symptoms of cystic fibrosis should go away, okay? So, that's the idea. Let me now talk about the practicalities of how you actually do it. So, the first way that I'm going to talk about in which you could actually do it is using retroviruses. Okay, so you use a retroviral vector, okay, to get this new gene for CFTR into the cell. Okay, now let me just tell you a little bit about retroviruses. So an example of a retrovirus, easily the most famous example of a retrovirus, is human immunodeficiency virus, HIV, the most feared viral disease of all, probably. Okay, now the ideal thing about retroviruses is that they implant their genome into human cells. So let me just draw a picture of one of them. Okay, so basically the outer portion, this outer line, which I'll colour in a specific colour, so this portion that I'm now colouring in blue here, this is a phospholipid bilayer that surrounds the viral particle, okay, and this is what's known as the viral envelope, okay, and it will have all sorts of integral membrane proteins within it. Then inside the viral envelope, this hexagon that I've drawn here, this is representing the capsid. Okay, and it doesn't necessarily look like a hexagon in the case of all retroviruses, okay, but I'll draw it as a hexagon nevertheless. Okay, so this is a capsid. Now, what is a capsid? A capsid is basically a shell that is made up of proteins. So those little proteins have come together to make a shell that is protecting the genome which is inside. Okay, now in the case of retroviruses, they have a genome that is RNA. Okay, so it's RNA genome. Now, basically, when they infect a cell, what happens is they use a special enzyme which they'll have within them, okay, which they'll bring with them, known as reverse transcriptase, to turn the RNA genome back into a piece of DNA. Okay, so reverse transcriptase synthesizes a piece of DNA that is complementary to the RNA, okay, and then what they can do is they can use a DNA-dependent DNA polymerase to then synthesize the other piece of DNA from that RNA. Okay, so if I show this, there's the RNA. What will happen is you'll take this piece of RNA, which I've zoomed in a bit, and the reverse transcriptase will produce a piece of DNA that is complementary to that. So there's the RNA in red, and the reverse transcriptase will produce a piece of DNA that is complementary to that. I'll just move this up. Okay, then what will happen is you'll get rid of the RNA here, and another enzyme will then synthesize the complementary piece of DNA to that piece of DNA that you produced firstly. Okay, so you'll then synthesize another piece of DNA. Okay, then what happens is this double-stranded piece of DNA that you now have gets implanted into the host cell's own genome. Now that's important. Okay, what happens is the retrovirus just cuts the DNA at some random point, and then will insert in its double-stranded piece of DNA here in vivid purple. Okay, like so. Okay, so this is the host genome with our double-stranded piece of DNA, which is the viral genome, now implanted in. Okay, now there's a special word for this double-stranded piece of DNA, which is the virus genome. It's called a provirus. Okay, right. So this is ideal because, basically, if we produce that piece of mRNA so that when, or RNA rather, so that when reverse transcriptase uses it and creates a provirus, this provirus contains the CFTR gene. Okay, so this is now going to contain the CFTR gene here. Okay, if we create that piece of I can't stop myself saying mRNA. If we create that piece of RNA perfectly correctly, okay, we can get it so that the piece of DNA that you synthesize from that contains the CFTR gene, which I'll highlight here in blue, and then what's going to happen is you're going to have this CFTR gene implanted into the host cell's genome. Okay, so the idea is that you could give this by inhalation. Okay, and that's one of the things that you have to be careful of. You can't just inject this into someone, okay, because it's most likely going to cause an immune response. Okay, so you have to instead give it by inhalation into the lungs. Okay, then what can happen is it can infect the cells of the respiratory epithelium, and at least into those cells of the respiratory epithelium it can put a new CFTR gene. Now, remember which cells had the problem in the respiratory epithelium. Okay, it was the cells of the acini right at the base of the submucosal glands, basically. So if I get this piece of paper back again, okay, I'll just try and find it. Is this it? Yes, here we are. Okay, remember, it's the cells of the acini here, okay, which no longer could secrete the serous secretion which was necessary for the mucin to be added to create mucus. Okay, and that created the thick, sticky mucus. Do you think these cells are going to be particularly accessible to a virus that is being inhaled? The answer is no. So that's the first problem we're starting to see here. And the other obvious problem is that if we can only give it by inhalation, then it's not going to help with the problems in the pancreas, it's not going to help with the problems in the intestine, it's not going to help with the problems with the sweat gland, and it's certainly not going to grow you vas deferens back again. Okay, so it's only going to help with the respiratory symptoms, but since those ones are the ones that kill you, that's not too big a problem. Okay, we can give you enzyme replacement to deal with the pancreatic insufficiency problems. Okay, but the main problem with even treating the respiratory symptoms is how do you get the virus to gain access to the acini when they are right at the base of the submucosal glands here. Okay, right. So now we're going to come on to the other problem of using retroviruses. Okay. Oh, actually, before I tell you about the other problem, I should probably tell you about the good side of this. The good thing about this is that if you can get this new piece of DNA into the stem cells which produce the cells that line the respiratory epithelium, then it will be a one-off treatment, basically, because if it's actually implanted into the host cell genome, then when the cell divides, then the daughter cells will also have this new piece of DNA in, okay? And therefore it will be a permanent treatment, it will be a cure, if you like. Okay, so as long as you get it into the stem cells which produce the other cells which line the epithelium, okay, then it's a permanent resolution to the problem. Okay, so that's the good thing about retroviruses implanting it into the host cell's genome. The problem with you implanting the new gene into the host cell's genome is that retroviruses just implant it in anywhere, basically. They do not pick an ideal place to put it in. Okay, they just go anywhere and put it in anywhere. So they could have cut through some other gene which could cause other problems, basically. Now, particularly, this can lead to the formation of cancer, okay? Because remember, cancer begins as mutations in genes which make you over-divide, okay? So for instance, if you insert your new piece of DNA right through a tumor suppressor gene, okay, which has its role in preventing the cell from over-dividing, then you're going to take out the function of that tumor suppressor gene, okay, and that means that the cell is potentially now going to over-divide, and you are starting the process towards the development of cancer, okay, so this is pro-cancer, basically, is the problem, okay, and that's one of the major problems with retroviral gene therapy, okay. I remember being told about the fact that another genetic disease, specifically severe combined immunodeficiency, SCID, okay, was treated with in vivo gene therapy using retroviruses. Okay, it's another disease caused by a mutation in a single protein. Okay, and they used retroviral in vivo gene therapy to treat it, and you know, it was all in the papers. They talked about how this boy who had previously had to live in this bubble all his life because of his extreme immunodeficiency could finally come out of the bubble now that he had been given this in vivo gene therapy. So it worked fantastically. The problem was that he then went on to develop cancer, specifically leukemia. Okay, so yes, there are problems with retroviral gene therapy, basically. Okay, it's pro-cancer. Okay, so there are ways around this, however. We can use a different type of vector. We can instead use adenoviral vectors rather than retroviral vectors. Now, what is the advantage of using an adenoviral vector? Well, basically, adenoviral vectors are slightly different from retroviral vectors, okay, in that instead of having an RNA genome, they have a DNA genome, okay? So it's a double-stranded piece of DNA, so I'll show this. So here is the double-stranded piece of DNA, and we of course can insert our healthy CFTR gene into this double-stranded piece of DNA. And the advantage to adenoviruses is that when they infect cells, they will put this double-stranded piece of DNA into the nucleus of the cell, but they will not implant it into the host cell genome, so they will not have the problems with causing cancer. Now, there are all the same issues with not being able to inject this into the blood, therefore you have to give it by inhalation, okay, aerosol inhalation, okay, so that's the exact same issues as we have with the retroviral vector. But now you lose the advantage that the retroviral vector had. Okay, so there are problems with this approach as well. Remember, there was an advantage to the retroviral vector implanting it into the genome, which is that if it implanted it into the genome of a stem cell, then all the progeny of that stem cell would then have this new gene. Okay, so it was a cure. It was a one-off treatment, and then you'd be cured. Okay, the problem with adenoviral vectors is this just implants it into the nucleus. So when the host cell divides, its daughter cells are not going to end up with this new piece of DNA. Okay, so basically, because the respiratory epithelium is continuously turning over, the cells are continuously being replaced, you can do this treatment, but, you know, after a year or so, very quickly, what's going to happen is the cells are going to be replaced, and then they're not going to have this new piece of DNA in anymore, and therefore you're going to have to redo the treatment. And what's the problem with redoing the treatment? Well, even if you're not giving the viral particles intravenously, even if you're just inhaling them, they're still going to promote an immune response. Okay, it's not going to be as big as if you'd injected it into the blood, but they're still going to be promoting an immune response. Okay, so every time you re-give the treatment, you'll be getting a stronger immune response, and you could induce basically something similar to an asthma attack in someone every time you give this treatment. So that's the problem, that because you have to re-deliver it, the body is going to mount an immune response against this viral particle, okay, and that can cause severe problems with being able to re-deliver it, okay, so you're going to get immune responses. Okay, right, now let me talk about the final type of vector which you can use to deliver gene therapy. Finally, there are liposomes which you can use. Okay, now what does a liposome consist of? A liposome is literally just a membrane, okay, so a spherical membrane, the surface of a sphere, okay, and then you're going to put your DNA inside. Now generally the DNA is put inside as a plasmid, okay, so a loop of double-stranded DNA like so. So here is our DNA plasmid, okay, and into this DNA plasmid we can put our CFTR gene. Okay, so let me show this. So we'll have this little blue bit as our CFTR gene here. Okay, now, what's the advantage to this then? Well, firstly, this does not trigger an immune response. Okay, so you can inject this intravenously and it can go to all the portions of the body affected by cystic fibrosis. Okay, secondly, it can be given multiple times without promoting an immune response. Okay, so that gets around this problem here, and it also gets around the problem that we were only able to give it by inhalation. Okay, right, what is the problem then with liposomes? Okay, well the problem is that they are not particularly good at delivering the DNA into the nucleus, okay, because all that's going to happen is this liposome is going to fuse with the membrane of cells. Okay, so if I show this, here is, let's say, a cell here. Okay, what will happen is the liposome will fuse with the membrane of the cell, and then it will just deliver the DNA plasmid into the cytoplasm, and then it's just luck as to whether that DNA plasmid actually gets into the nucleus, where there is the machinery for transcription of that DNA. Okay, so liposomes are extremely inefficient, which is the problems that they have. Okay, so it's very unlikely that they'll actually deliver the CFTR gene to where they need to go. Okay, so that's the big problem with liposomes, that they don't actually deliver it into the nucleus. And of course they have the same problem as the adenoviral vectors, that because they don't implant it into the genome, then it's not a permanent change. You have to continuously re-deliver the gene therapy. So although there was great hope for in vivo gene therapy and the potential to cure cystic fibrosis with in vivo gene therapy, at the moment it is not working. It has not worked. It's not currently something that I think is clinically used at all, okay, because of the dangers of retroviral vectors, and then the problems with the immune response upon recurrent delivery with adenoviral vectors, and then the huge inefficiency with liposomes, and the reality is that they don't really work, basically, is the problem with the liposomes. Okay, so it's something that may well develop into a future treatment, but at the moment it's not currently realised because of the problems with it. Okay, right, now what I'm going to talk about is drugs which can be used to treat specific mutations in CFTR, okay, and can help you actually acquire some function of CFTR back again. Okay, so we're going to start off with drugs that can be used to treat class 1 mutations in CFTR. Okay, so remember our example of a class 1 mutation in CFTR was R553X. Okay, so remember what that meant is you had taken the arginine, okay, and let me just draw this out, the arginine at position 553, so here's our arginine at position 553, and you had changed the codon for arginine at 553 to a stop codon. Okay, so now what was happening is the protein was getting prematurely stopped when it was being synthesized by the ribosome. Okay, so how can we treat this? You might think, well, how on earth can you treat that? Well, actually, there is a drug that is used to treat these sort of class 1 mutations, okay, and it's known as gentamicin. Okay, now gentamicin is actually an antibiotic within the class of aminoglycosides. Okay, now what aminoglycosides do is they bind to ribosomes in bacterial cells, and that's how they exert their effects. Okay, however, if you give gentamicin in human cells, it also binds to ribosomes. Okay, and what it can actually end up doing, and why it's useful in class 1 mutations in CFTR, is that it actually can promote ribosomes skipping codons. Okay, now of course this will be completely at random, so let me just explain what I mean by this. So let's have our ribosome here. Okay, and of course what our ribosome will be doing is it will be working its way along an mRNA here. Okay, and it will be looking at each codon in turn, and I should have just... I was drawing that far too big. Okay, it will be looking at each codon in turn, so for instance this codon here, and putting an amino acid in for that codon. Okay, now, what we would absolutely love the ribosome to do is skip codon at position 553. Okay, so we would love it to skip the codon that codes for the amino acid at position 553, because if it could skip that codon, remember, has been turned into a premature stop codon, and that's causing the huge problem. Okay, that's the reason that we're not getting any functional CFTR produced, because we're stopping prematurely. Okay, if we could skip that codon and just move on to the next one and put on that as the next amino acid, then we'd produce a CFTR that was missing one amino acid. It would miss this arginine, but you know, that's better potentially than a prematurely stopped protein. Okay, yes, delta F508 wasn't really that great either, and that was one amino acid lost, but potentially R553, delta R553 wouldn't misfold as badly as delta F508, and therefore would make it to the apical membrane. So what gentamicin does is it binds to the ribosome. Okay, so I'll draw the drug molecule here. This is gentamicin. It binds to the ribosome. Okay, so this is the drug molecule, and I'll colour it in vivid purple. And it does promote the ribosome just skipping codons. Now this skipping of codons will be random, okay, but occasionally what will happen is the ribosome will end up skipping that stop codon at position 553, and therefore you will get a protein that is nearly the same as the original CFTR, but the fact that it's got one amino acid missing. And if you're lucky, that one amino acid missing won't make too much difference, and therefore it will end up going to the apical membrane, and therefore you will end up with some functional CFTR being produced from this gene that had this class 1 mutation in it. Okay, so that's a drug that can be used to treat class 1 mutations in CFTR and try and bring a bit of function back to the CFTR with that class 1 mutation in it. Okay, right. Next, what I want to talk about is treatments for class 2 mutations. And our prototypical mutation that is a class 2 mutation is delta F508, which causes the protein to misfold, remember. Okay, so treatments that can be used to help here are chemical chaperones. Okay, so what are chemical chaperones? Well, chaperone is anything which helps fold proteins. Anything which helps to a protein to fold is called a chaperone. Okay, so chemical chaperones are small molecules, small drug molecules, rather than huge, great proteins, which can help proteins to fold. So examples which can be used are firstly glycerol, okay, and then also phenylbutyrate. Okay, so these are examples of chemical chaperones which can be given if you've got a class 2 mutation and can help the mutated CFTR protein to fold correctly. Okay, so let me just show you the structure of phenylbutyrate. I hope you all know the structure of glycerol, but the structure of phenylbutyrate basically is butyric acid, or the conjugate base of butyric acid, with a benzene ring attached. So I'll start with the benzene ring here. I'll just move this up first. Okay, so this is our benzene ring here. Okay, drawn in its skeletal form. And then I'll draw the butyrate in its molecular form. So butyrate is a 4-carbon carboxylic acid. It's the old name for butanoate, okay, as it would now be called. Okay, and then here is the carboxylic acid group. Now, butyrate specifically means the conjugate base of butyric acid, so this oxygen here should have a negative charge rather than having the proton attached there. If I had written phenyl butyric acid, okay, that would have meant the actual acid molecule where you've still got the proton here. Okay, butyrate is what's known as the conjugate base of butyric acid. Okay, so once butyric acid has lost its proton from this alcohol group of the carboxylic acid group, it can no longer donate another proton away. Okay, so it's no longer an acid molecule, because remember the definition of an acid molecule is something which can donate a proton away. This is not capable of donating a proton away into solution. Okay, in fact, it's actually a base molecule because it's capable of accepting a proton back out of solution. A proton can join on to that negatively charged oxygen. Okay, hence why it's called the conjugate base of butyric acid. Okay, right. Now, of course, you can't just give a molecule that's got a negative charge like this, okay? You can't give a pill that's got a massive, great negative charge. So instead, what you actually give is a salt of phenyl butyrate, and usually the salt that's given is sodium phenyl butyrate, where the sodium is bound to the negative charge of the oxygen. Okay, so often what's given is actually sodium phenyl butyrate. Okay, and that's designed, well, that's supposed to help the mutant CFTR protein to successfully fold into the correct shape, and therefore hopefully it won't end up being destroyed by the cell's quality control mechanisms. And then hopefully some will actually end up getting to the apical surface of the cells, and therefore you might restore some function of the CFTR back to those cells. Okay? Right, now the final drug that I want to talk about, which can be used to treat both class 2 mutations and also class 3 mutations. So first I'll just remind you of class 3 mutations. Okay, so remember class 3 mutations such as G551D, okay, were mutations which resulted in a CFTR that was very difficult to activate, or which barely activated at all. Okay, so remember, this was a missense mutation which resulted in glycine at position 551 being replaced by aspartic acid. Okay, and that was in the nucleotide binding domain number 1 of the cystic fibrosis transmembrane conductance regulator, and reduced the ability of ATP to activate the CFTR. Okay, right. So now the drug that I'm going to talk about is a drug that I think is still in clinical trials at present, okay, but it's looking hopeful. Okay, it might be available clinically by now, I don't know. Okay, it's a drug called ivacaftor. Okay, now how does ivacaftor work? Well, it's what's known as a CFTR, Cystic Fibrosis Transmembrane Conductance Regulator, potentiator. Okay, now what does that mean? Basically, this is a drug which will bind to CFTR and will increase the time for which it is open. Okay, so let me just go back to my original drawing of CFTR. Okay, so here we are. Remember how CFTR works. Sometimes it is closed, sometimes it is open. So we start off with it closed. Okay, in order to even begin the cycle of opening and closing, you need a phosphorylation of the regulatory domain by protein kinase A. Okay, then what can happen is a single ATP can bind to nucleotide binding domain number 1, and that causes nucleotide binding domain number 2 to come and bind to the nucleotide binding domain number 1 with the ATP bound. Okay, so you get dimerization of NBD1 with NBD2. That is still closed. Okay, then once NBD1 has dimerized with NBD2, that means that now another ATP can come and bind to NBD2, and then that causes a change in conformation, specifically in TMD1 and TMD2, which then causes the thing to turn into the open state whilst it's in which it's conducting chloride anions across the cell membrane. Then what will happen is this ATP that's bound to NBD2 will be hydrolyzed down to ADP and an inorganic phosphate, which will just be chucked off. So this is the inorganic phosphate here. Okay, and that's still in the open state as well. Then what will happen is when this ADP finally cleaves off, then you'll be back in the state where you've got an ATP bound to NBD1 and NBD2 bound to the NBD1 with the ATP. Okay, and that's then a closed state, so it will go back into the closed state. Ivacaftor basically binds to the cystic fibrosis transmembrane conductance regulator and keeps it in the open state for longer. So when it goes through one of these opening cycles, it will stay in the open state for longer, and it will take longer to go back into the closed state. So when the CFTR opens, it will open for longer when ivacaftor is bound to it. Okay? Right. So, what does that mean then for delta F508 and G551D? So let's start with why that's useful for delta F508. So remember, in delta F508, you've got this misfolded CFTR which mainly gets destroyed by the cell's quality control mechanisms. Now, the cell's quality control mechanisms are not perfect. Some misfolded CFTR is missed, or either some CFTR might fold in such a way that it doesn't get picked up by the cell's quality control mechanisms as faulty in the first place. The point is that even if you've got two mutations in in CFTR that are delta F508 mutations, you will still get some CFTR going to the apical membrane. It's just far too little, basically, okay, to actually make a reasonable contribution. Okay, so the idea is, those CFTRs that do get to the apical membrane and do function to some extent, even with the delta F508 mutation, they are still reasonably functional. Okay, and that's why it's such a shame that the cell's quality control mechanism bins so many CFTRs with the delta F508 mutation, because the reality is when they get to the apical membrane, they are reasonably functional. Okay. Regardless, those CFTRs with the delta F508 mutation that do get to the apical cell membrane, if we give the ivacaftor drug, these will now remain open for longer, and therefore the actual function of them is going to be increased. Okay, so we are increasing the function of those CFTRs which actually do get to the apical membrane. Similarly, with G551D, okay, now our CFTR proteins are really, really difficult to actually activate, but some of them will eventually activate, okay? It's just they're really, really difficult to activate. And those ones that, when they do activate, those will now be open for longer in the presence of this ivacaftor CFTR potentiator, and therefore the function of CFTR, the overall chloride conductance, will have increased in the presence of this drug. So this drug basically increases the amount of chloride that is moving through the CFTR activity that is present, basically. Okay, so it potentiates the activity of CFTR that is present even with these mutations in CFTR. Okay, right, so that now concludes our discussion on cystic fibrosis and cystic fibrosis medications.